Okay, so welcome to your review for Advanced. Our first basic brain review exam is coming up, and if you have any questions, please email me about them before the exam actually is administered. But I want to go over a few topics actually that relate to this exam. So on the website are some explicit instructions about certain topics to go over and topics to review. Please be serious and go through those because the questions will relate pretty directly to those topics. Your teaching assistant, your very able teaching assistant, will also help you work through some of these topics in class. All of the functions of the macroglia, we went over the functions of the astrocytes, which are macro or large glia, and also the myelinating glia. Remember the Schwann cells that you find in the periphery and the oligodendrocytes that you find in the central nervous system. They have a number of distinct functions, those macroglia or large-scale glia versus the microglia, which really are serve to basically attack invading microorganisms and potentially help modify synaptic connections. Remember, some of the details of the resting and action potential. Know, what that voltage line looks like. And remember, you have depolarization, and then you hit threshold, and then the voltage-gated sodium channels open. The sodium rushes in, you get the voltage line going down, and then, you've got the potassium channels opening as well, and the potassium is rushing out, and that line is dropping, and at the same time, the sodium is moving away down the axon. That brings things back to a repolarized resting potential state, right? Definitely know about the different kinds of neurons that were discussed in that introductory chapter that we read. For example, like unipolar or pseudo-unipolar neurons, like the dorsal root ganglion neurons, bipolar neurons, like the ones you find in the retina, for example, or actually the olfactory neurons in the nose or bipolar cells. Then the most common type of CNS interneuron you'll find, right, is the multipolar cell with lots of projections, both at the input or dendritic end and the output or axonal end. Know about, let's see, how complex a lot of what's discussed in intro actually is. For example, how we have different classes of receptor that we introduce in introductory neuroscience classes like metabotropic or ionotropic, and yet there are additional classes, and there's a lot of combinations of different drivers of receptor activity. For example, with glutamate, which is our primary excitatory neurotransmitter, remember it binds to ionotropic receptors that open up sodium channels, right? So you can depolarize and get excitation, right? Reach threshold and initiate an action potential. However, there's a certain kind of glutamate receptor. It's called the NMDA receptor, and that won't just open in response to the binding of the neurotransmitter. It's not like a classic ligand-gated channel or ionotropic receptor. It requires a lot of activity, a lot of excitatory activity of that synapse. So you've got to actually get lots and lots of depolarization in addition to the binding of the ligand. So it's more complicated. That receptor isn't just ligand-gated, it's also partly voltage-gated. So there's a combination of all these different kinds of interactions around receptors. So it's much more complicated than we initially discussed. Know the difference, that was brought up in the Mountcastle paper between sort of primary sensory and motor regions of neocortex. Those we refer to as heterotypic, regions. And we talk about them as being heterotypic because hetero means different. So they're very, very different from the other regions of neocortex, which, is referred to as homotypical. They're more alike in that they merge multiple, sensory and motor planning kinds of inputs. We sometimes call those homotypical regions the areas that are much more common in humans versus other primates, for example, as association areas of neocortex. And in heterotypic regions, they're also kind of different in that we sort of know how the columns, and by this I mean, right, combinations of mini columns that serve a functional purpose that, are connected. These are neurons that are connected across the neocortex, across those 6 layers, but they respond to some particular kind of stimulus. So, primary visual cortex, for example, that's definitely a region of heterotypic neocortex. It responds, the columns, the sort of macro columns or functional columns, respond to bars of light that are oriented in particular, orientations in specific regions of visual space, right? It's much more complex to figure out, like, what functionally organizes those larger columns, again, made up of the individual mini columns. That's the basic structural unit of the neocortex, right, which is a result of, developmental processes, the radial glial cells going out, right, and the neurons all following their way out. But it's harder to figure out how many mini columns form a functional column in, like, parts of the frontal lobe, for example, and what functionally drives those macro columns, those individual columns, right, in an area like, I don't know, anterior cingulate or, Brodmann area 9 or something like that, right? Definitely consider Ohm's law. We talk about that a little bit in the introductory chapter. Voltage equals current times resistance. And understand that if we take the voltage down, right, to threshold, if we depolarize the threshold, well, there's going to be a significant change in the resistance of the membrane because you're going to suddenly open all of those voltage-gated sodium channels at minus 55. And when you reduce the resistance, right, you increase the current. That's the relationship, V equals IR. So, the resistance is high at the resting potential at minus 65, right, because there are no or very few leakage channels, very few channels open to sodium. So the resistance is high and the current is low. But if you drop the resistance, well, the current goes up, and that's your action potential. That's the initiation of the action potential, the sodium current rushing along. We talked about en passant terminals, right? They're like kind of like a normal axon terminal, right, with synaptic vesicles and all the release machinery, for release of neurotransmitter, but they are not located at the end of the axon, right? They're actually located in these swellings along the length of the axon. And a very good example of that is the locus ceruleus, which releases norepinephrine. So, bam, when there's I have a dog over here that just leaped. When you have a startle response, you get, release of norepinephrine, from these locus cerulear neurons, which actually have these swellings all along their length, where you find, the vesicles, filled with neurotransmitter, the voltage-gated calcium channels, that couple the depolarization of the action potential to, the entry of calcium, and then the fusion of the vesicle and the release of neurotransmitter into the cleft. So you can, one single neuron can have a wide effect on many, many synapses with these en passant, in passing, kind of terminals or terminal swellings. And we talk a little bit about why EPSPs and IPSPs that you record postsynaptically when you're recording the electrical activity in response to very tiny stimulations of the presynaptic, terminal, actually don't kind of rise or fall in a very a graded way, but rather they actually step. They're like multiples of a specific unit, a quantal unit, right? And that's due to the fact that the smallest amount of release you can get presynaptically is one vesicle of neurotransmitter. So that's going to lead to, like, let's say 20% of the neurotransmitter in that vesicle will reach and stimulate the ionotropic receptors on the next cell. And so that's not going to be, you can't just get one neurotransmitter. You're going to go step one vesicle, two vesicles, three vesicles, two vesicles, 8 vesicles. It'll be some multiple, because of the vesicular nature of neurotransmitter release. All right, so know some basic brain regions. Know that, for example, you got, here's your brain, right? When you get sensory information, heading towards areas of cortex, for example, there's initial processing, like in a nucleus in the thalamus. Know the superior colliculi are important for visual orienting. Know some of the basic neuron. Know the basal ganglia are critical for movement. Some of the basic things we discussed in the intro class, and we've been alluding to in our discussions of these articles, all the way through. Definitely know the default network. We talked about a lot, about default and about what it's important for. That it's critical for rumination, for autobiographical memory, for taking the perspective of others, for planning and playing out various scenarios. So it's a very important structural network, and it has very specific nodal components. So we talked about, for example, rich nodes, right? Including the posterior cingulate cortex, a very critical node in this network. The anterior cingulate cortex is also a critical node in this, distributed default network. Mostly though, in the anterior cingulate, it's interesting because it's primarily sort of, more medial or ventral regions, while the more dorsal region of the anterior cingulate is engaged when you are, engaging your selective attention network, where you have more, lateral and dorsal regions on the side of the brain. So remember, for selective attention, right? You have, posterior parietal cortex, right? You've got dorsolateral prefrontal cortex, and you have anterior cingulate that form a network that seems to be engaged when you're attending visually, to things in your environment. While when you shift internally, and ruminate about something and think about your past and plan for the future, you actually go more medial and ventral. So it's more medial ventral, or it's more ventral anterior cingulate. It's definitely posterior cingulate. There's a lateral component of the default network. It's called the inferior parietal cortex, or the inferior parietal lobule, but it's also more ventral, versus the posterior parietal cortex. So there is definitely, with default, when you shift to default, there's a shift from more dorsal and lateral, side regions of the cortex that are networked together, to more medial and ventral, regions of the neocortex. Think about how default, when you are actually engaged in sort of memory, like, recalling explicit memories, there seems to be engagement of the medial temporal lobe. So that's also considered part of the default network. It's a subsystem that interacts, with these core regions like the posterior cingulate, anterior cingulate, inferior parietal lobule, when you are engaged specifically in sort of recalling, explicit memories from your past. Okay, so know the techniques. So, one of the ways we know that default is a default network, right, is because we've used something called resting state functional connectivity, MRI, right? Which means basically you look at, blood flow, changes, and remember these techniques focus on, the bold signal, right? The how much deoxygenated hemoglobin is in a region, right? And when you utilize a region, you actually get increased blood flow into that region, and you basically see more oxygenated blood come in, and that can be, that difference or change can be utilized to identify which areas are more active, right? Because that follows on changes in activity, actual neuronal activity. And so if you look at tiny little fluctuations in blood flow in different areas of the brain, you see some areas are significantly correlated, and those areas that are correlated, are changing their activity in concert. So they're sort of communicating with each other. It's an indication that they are functionally connected. And we see, definitely using this form of resting state functional connectivity, MRI, that posterior cingulate is connected to anterior cingulate, which is connected to the inferior parietal lobule, which is connected to the medial temporal lobe functionally. So these are functional connections. You can also use, for example, MRI and focus on the movement of water, right? We talked about that, right? It's called diffusion tensor imaging, and where water is able to diffuse in a specific, manner, it can't go anywhere. It has to go in a particular pathway. Well, that's going to be indicative of white matter in that particular region. So this is a structural technique that can be used to say, okay, these areas are functionally connected, right? So are they actually physically linked to each other, by white matter connections? So that's a really good way to look at that. So no, CAT scan uses X-rays, no PET scan looks at the decay of radioactive material. Go over some basic techniques, because we did discuss that quite a bit. I think you will do very well on this test. Don't panic, but definitely go over this material that I'm kind of emphasizing here today. Ask Bradley questions, or you ask your teaching assistant questions, and send me any questions that you have as well. Okay, good luck, and I will see you.